This lecture is on nervous tissue. What is the nervous system? The nervous system is the master control system of the body. Nervous system functions include sensory input, integration, and motor output. Sensory input occurs when receptors are stimulated and send information to the brain and spinal cord. Integration occurs when the brain and spinal cord process the sensory information and determine what response is necessary. Motor output occurs when motor neurons send signals to muscles and glands, which are called effectors. This illustration shows sensory input, integration, and motor output. Sensory input occurs when you look at a glass of water while you are feeling the sensation of thirst. That visual cue, as well as the sensation of thirst, will be integrated in the control center, which is the brain and spinal cord. Motor output occurs when the brain has made a decision to engage the effector organ, or the muscle that will help you to grasp the glass of water and lift it towards your mouth, and then the muscles that will help you to drink and swallow the water. There are two major divisions to the nervous system: the central nervous system and the peripheral nervous system. The central nervous system, abbreviated CNS, includes the brain and spinal cord. It functions in integration. The peripheral nervous system, abbreviated PNS, includes the nerves and ganglia. It functions in sensory input and motor output. This illustration shows the central nervous system in gray, containing the brain and spinal cord. The peripheral nervous system is in tan and contains the peripheral nerves and their ganglia. The sensory division of the peripheral nervous system carries information towards the central nervous system. Somatic sensations are typically perceived consciously. They arise from the skin, skeletal muscles, and special sensory organs. Somatic sensations include touch, temperature, proprioception or balance, sight, taste, smell, and hearing. Visceral sensations are typically not perceived consciously. Visceral sensations arise from the internal body organs, smooth muscles, cardiac muscles, and glands. Visceral sensations include pain, bloating, and distension. Generally, when you are feeling visceral sensations, they are not pleasant. The motor division carries information away from the central nervous system. Somatic motor effectors are typically voluntary and are controlled consciously. Somatic effectors include the skeletal muscles. Autonomic motor effectors are typically involuntary and are controlled without conscious input. Autonomic effectors include the cardiac muscles, smooth muscles, and glands. The autonomic nervous system, abbreviated ANS, is divided into the parasympathetic nervous system and the sympathetic nervous system. The parasympathetic nervous system maintains homeostasis. The sympathetic nervous system maintains blood pressure and performs the stress response. This illustration shows the different types of divisions of the peripheral nervous system. The sensory nervous system is involved in the input. It detects stimuli and transmits information from receptors to the central nervous system. Somatic sensory input is sensory input that is consciously perceived from receptors, such as the eyes, ears, and skin. Visceral sensory input is sensory input that is not consciously perceived from receptors. These include the blood vessels and the internal organs. Once the information is processed and integrated in the control center, then the output belongs to the motor nervous system division of the peripheral nervous system. The motor nervous system initiates and transmits information from the central nervous system to the effectors. In the somatic motor division, motor output is consciously or voluntarily controlled, and the effector is the skeletal muscle. In the autonomic motor division, motor output is not consciously or it is involuntarily controlled. Effectors are cardiac muscle, smooth muscle, and glands. The central nervous system integrates sensory input and motor output based on other sensations, memories, emotions, learning, and reflexes. This illustration shows an example of specialization and integration. We are going to see the brain areas that are active when hearing, seeing, and speaking words. When hearing words, the auditory cortex and the Wernicke's area light up. When seeing words, the visual cortex and the angular gyrus light up. When speaking words, the Broca's area and the motor cortex light up. This is an example of integration, and I know you're all thinking it, so I'll say it. Hear no evil, see no evil, speak no evil. Nervous system functions resemble the homeostatic feedback loop. The nervous system is integral to maintaining homeostasis. This illustration shows the homeostatic feedback loop we learned about back in Chapter 1. The first event is the stimulus producing a change in the variable, and the variable is anything that should be kept in balance within your body. For instance, thirst. Thirst will be our stimulus, and our receptors are going to detect that change. And the receptors may be a drying out of the epithelia in our mouths. As the information travels towards the control center, it goes through step 3, input. Information is sent along the afferent pathway. Afferent approaches. It's the sensory pathway towards the control center. The hypothalamus is the center in the brain that regulates thirst. And thus, the output from the control center will go towards the effector organs that will then initiate the last step, which is the response. And that will be to lift a glass of water and take a drink. Now let's look at the gross anatomy of nervous tissue. We'll start with the gross anatomy of nerves and ganglia. Nerves are cord-like organs found within the peripheral nervous system that contain bundles of axons, along with blood vessels and connective tissue. Nerves are actually going to be organized much like muscular tissue. Most nerves are mixed, meaning sensory and motor axons travel alongside each other within the same nerve. Axons are insulated from each other by layers of connective tissue. This become the nerves here, this split. In the spinal cord, we have areas that receive sensory input. These are the dorsal horns. And then we have areas that send out motor output. These are the ventral horns. Roots connect the nerves to the spinal cord. The dorsal root will contain sensory neurons that are approaching the spinal cord. The ventral root will contain motor neurons that exit the spinal cord. The spinal nerve itself is mixed. The sensory and motor axons join, and then they can branch and follow along into major nerves that innervate muscles and glands in the body. Layers of connective tissue in a nerve include the epineurium, perineurium, and endoneurium. The epineurium surrounds the entire nerve. It protects the nerve like a leather sheath. The perineurium surrounds fascicles of axons. It is a fibrous sleeve that protects and supports bundles of axons. Endoneurium surrounds individual axons. It contributes to electrical insulation of axons. The epineurium and perineurium are composed of dense, irregular connective tissue. The endoneurium is composed of areolar connective tissue. All three connective tissue layers contain blood vessels and lymphatic vessels. As you can see, this is a very similar arrangement as seen in a muscle. Here is a nerve showing the epineurium, the perineurium, and the endoneurium. The epineurium surrounds the entire nerve and protects the nerve like a tough leather sheath. The perineurium surrounds a bundle of axons, and the endoneurium surrounds individual axons. This helps to insulate the axons from one another, because recall, we have sensory and motor axons traveling in the same nerve. There are two major types of nerves, cranial nerves and spinal nerves. Cranial nerves attach to the brain. There are 12 pairs of cranial nerves. Cranial nerves can be sensory only, motor only, or mixed. Most cranial nerves innervate the head and neck and receive the special senses of sight, hearing, taste, and smell. Spinal nerves attach to the spinal cord. There are 31 pairs of spinal nerves. All spinal nerves are mixed, both sensory and motor. Many spinal nerves combine to form networks called plexuses that innervate the upper and lower limbs. This illustration shows the cranial nerves. There are 12 of them, and we will learn more about them as we move through the nervous system lectures. Here are the spinal nerves. There are 31 pairs of spinal nerves, and they are named based on the region of the spinal cord and the vertebral column that they arise from: cervical, thoracic, lumbar, sacral, coccygeal. We will also talk about spinal nerves in more detail as we move forward in our lectures. Ganglia appear as swellings on cranial and spinal nerves. There are sensory ganglia and motor ganglia. Sensory ganglia contain the cell bodies of sensory neurons. Ganglia of the autonomic nervous system contain synapses between autonomic motor neurons. The illustration on the left shows a sensory ganglia. It is close to the spinal cord and contains the cell body of the sensory neuron. The autonomic nervous system contains motor ganglia that connect a preganglionic neuron to a postganglionic neuron. These ganglia are located in different areas of the body based on whether they belong to the parasympathetic nervous system or the sympathetic nervous system. Let's look at the microscopic anatomy of neurons and neuroglia. Nervous tissue includes neurons and neuroglial cells. Neurons are excitable cells that conduct impulses. They are the functional cells of the nervous system. Neuroglial cells support the function of the neurons. The illustration on the left shows a neuron surrounded by numerous neuroglial cells. There are more neuroglial cells in the nervous system than there are neurons. The illustration on the right shows an artist's depiction of the neurons surrounded by the many different types of neuroglial cells. Neurons are the functional cells of the nervous tissue. The characteristics of neurons include excitability, conductivity, secretion, extreme longevity, and being amitotic. Excitability is the ability to respond to a stimulus. Conductivity is the ability of an electrical signal to travel along the length of the cell membrane. This is known as a nerve impulse. Secretion is the ability to synthesize and secrete neurotransmitters. Extreme longevity means that neurons can live for greater than 100 years. And amitotic means that neurons cannot undergo mitosis. It's a good thing that they can live for a long time because they do not divide and increase their numbers throughout your lifespan. Neurons come in many shapes and sizes, but generally consist of a cell body connected to one or more processes. The cell body consists of the nucleus and cytoplasmic organelles. Processes extend from the cell body. Processes include dendrites and axons. Dendrites are short and conduct impulses towards the cell body. The single axon is long and conducts impulses away from the cell body. An axon can branch as it travels towards other neurons, muscles, or glands. This illustration shows the neuron cell body and its many processes. Dendrites bring information towards the cell body. The axon carries information away from the cell body. The axon can branch in order to connect to several muscles, glands, or other neurons. Neurons are classified based on their structure and their function. Neuron structure refers to the number of processes extending from the cell body. Neuron function refers to whether the neuron receives sensory input, performs integration, or conducts motor output. There are 3 structural classes of neurons, each with specific functions. Multipolar neurons have many processes and are integration or motor in their function. Bipolar neurons have 2 processes and are sensory in their function. Unipolar neurons have 1 process and are sensory in their function. Multipolar neurons have many dendrites and a single axon connecting to the cell body. They are the most common type of neuron. They form the interneurons of the central nervous system and the motor neurons of the peripheral nervous system. 99% of neurons are interneurons, and they perform the function of integration. Motor neurons conduct motor signals to muscles and glands, and they include somatic and autonomic motor neurons. Somatic motor neurons connect to skeletal muscles, and autonomic motor neurons connect to cardiac muscles, smooth muscles, and glands. Bipolar neurons have 1 dendrite and 1 axon connected to the cell body. They are rare and are found in the retina of the eye and the epithelia of the nose. They are responsible for the sensory input for sight and smell. Unipolar neurons have a cell body that sits on a small process connected to a long axon. Unipolar neurons make up the majority of sensory neurons of the peripheral nervous system. They include somatic and visceral sensory neurons. They are found in both cranial and spinal nerves, and their cell bodies are found in ganglia. Somatic sensory neurons will receive sensory input from the skin and the special sensory the special sensory organs such as the eyes, and ears, and taste. And the skin will be touch, temperature, and pain. Somatic sensory receptors also receive information from proprioception or balance in your muscles. Visceral sensory neurons will receive information from visceral organs, such as the heart, the lungs, and the digestive viscera. This illustration shows sensory input from skin receptors. So this would be a somatic sensory neuron. It is a unipolar neuron. It has a cell body attached to one process. The sensory neuron cell body is housed in a ganglion. The terminal end of the sensory neuron enters the spinal cord. It synapses with an interneuron. Interneurons are multipolar neurons, and they make up 99% of the neurons in the nervous system, and they function in integration. So in this case, it can detect touch, temperature, or pain. Motor neurons carry the information from the central nervous system towards the effector. They are multipolar neurons as well, with many processes attached to the cell body. This is a somatic motor neuron because it is going to innervate the skeletal muscles. Neuroglial cells are supporting cells of the nervous tissue. They outnumber neurons, do not transmit nerve signals, are smaller than neurons, and can divide by mitosis. Neuroglial cells occur in the central nervous system and peripheral nervous system. Central nervous system neuroglial cells include astrocytes, ependymal cells, microglia, and oligodendrocytes. Peripheral nervous system neuroglial cells include satellite cells and neurolemmacytes. Astrocytes are the most abundant neuroglial cell of the central nervous system. Functions of astrocytes include the formation of the blood-brain barrier, regulating tissue fluid composition, forming structural support, assisting neuronal development, and occupying the space of dying neurons. Astrocytes help form the blood-brain barrier, which controls movement of materials from the blood to neurons. Astrocytes regulate tissue fluid composition by maintaining a constant potassium ion concentration. Astrocytes form structural support by providing a neural framework. Astrocytes assist neuronal development by directing fetal neurons and continuing to aid in forming synapses in mature neurons. And astrocytes occupy the space of dying neurons since neurons do not replicate. This illustration shows an astrocyte. Astro means star, cyte means cell. This is a star-shaped cell. It has many processes coming off of its cell body as well. Here we see the astrocyte forming the blood-brain barrier, and it does this by wrapping its cell membrane around blood vessel capillaries. This regulates the flow of nutrients and gases from the bloodstream towards the neurons. And you can see that the astrocyte is also connected to the neuron. It will absorb nutrients and gases from the blood and secrete the necessary nutrients and gases to the neuron. Astrocytes also form the structural framework of the nervous tissue, thus providing support for the neurons. Ependymal cells are ciliated, simple cuboidal, or simple columnar epithelial cells. They line the ventricles of the brain and the central cavity of the spinal cord. Ependymal cells that are entwined with capillaries form the choroid plexus. This secretes cerebrospinal fluid, abbreviated CSF. The ependymal cells also circulate the cerebrospinal fluid. This illustration shows ependymal cells anchored to the brain or spinal cord tissue. They have cilia that will help to circulate the cerebrospinal fluid. They also, along with capillaries, secrete the cerebrospinal fluid. Microglial cells are small macrophages that defend against pathogens in the central nervous system. Microglial cells replicate in response to infections, inflammation, trauma, and tumors. They are important because immune system cells are denied access due to the blood-brain barrier. They're called microglia because they're very small. So they're small glial cells. Oligodendrocytes are large cells with extensions that secrete myelin sheath around central nervous system axons. Myelin is a lipid that insulates axons so that nerve impulses are conducted quickly and efficiently. Oligo means scanty or few, and dendro means branches. So these are cells with a few branches. And as you can see, it helps them to myelinate multiple neurons within the central nervous system. This illustration shows the neuroglial cells just mentioned in the central nervous system. The astrocytes are pictured in green. The ependymal cells are in purple. The microglial cells and oligodendrocytes are in blue. The astrocyte functions are to help form the blood-brain barrier, regulate interstitial fluid composition by maintaining a constant supply of potassium ions, providing structural support and organization to the central nervous system, assisting with neuronal development, and replicating to occupy the space of dying neurons. Ependymal cell functions include lining ventricles of the brain and central canal of spinal cord, and assisting in the production and circulation of cerebrospinal fluid. Microglial cell functions include being phagocytic cells that move through the central nervous system and protecting the central nervous system by engulfing infectious agents and other potentially harmful substances. Oligodendrocyte functions include myelinating and insulating central nervous system axons and allowing faster action potential propagation along the axons of the central nervous system. Satellite cells are flat and surround the sensory cell bodies in the peripheral nervous system ganglia. They electrically insulate the cell body and regulate the exchange of nutrients and waste products between neurons and the extracellular fluid. Functionally, they are similar to the astrocytes of the central nervous system. This illustration shows the satellite cells surrounding the neuron cell body of a sensory neuron in the peripheral nervous system. Neurolemmacytes, also called Schwann cells, secrete myelin sheath around the peripheral nervous system axons. They are elongated and flattened, wrapping around the axons in a process called myelination. Myelin is a lipid that insulates axons and speeds the conduction of neuronal impulses. So these are similar to the oligodendrocytes of the central nervous system. The neurolemmacytes form the myelin sheath of the peripheral nervous system. This shows the process of myelination by a neurolemmacyte. In step 1, the neurolemmacyte starts to wrap around a 1 millimeter length portion of an axon. In step 2, the neurolemmacyte cytoplasm and plasma membrane begin to form consecutive layers around the axon as wrapping continues. In step 3, the overlapping inner layers of the neurolemmacyte plasma membrane form the myelin sheath. In step 4, eventually, the neurolemmacyte cytoplasm and the nucleus are pushed to the periphery of the cell to form the neurolemma. This illustration shows the neuroglial cells of the peripheral nervous system. The satellite cells shown here in red and the neurolemmacytes shown in blue. Satellite cell functions include electrically insulating peripheral nervous system cell bodies, regulating nutrient and waste exchange for cell bodies in the ganglia. Neurolemmacyte functions include myelinating and insulating the peripheral nervous system axons, allowing for faster propagation along an axon. The axons of the peripheral nervous system are vulnerable to injury. A damaged axon is more likely to regenerate if the damage is not extensive. The cell body and neurolemmacytes must survive. Chance of recovery is improved with a shorter distance to the site of innervation. The proximal portion of the area seals off. The portion distal to the injury degenerates in a process called Wallerian degeneration. Macrophages come in to clean the debris. Neurolemmacytes secrete growth factors and form a regeneration tube. The new axon will sprout from the proximal sealed end. An innervation is restored when the newly formed axon reaches its sensory receptor or its effector organ. This illustration shows the process of regeneration of a nerve in the peripheral nervous system. The cell body and neurolemmacytes must survive. The closer the injury is to the point of innervation, the more likely the success of regeneration. In step 1, distal to the injury site, the axon and myelin degenerate and fragment in a process called Wallerian degeneration. In step 2, Schwann cells proliferate along the original axon path. Macrophages remove debris. In step 3, the axon grows into the injury site along the Schwann cell pathway. In step 4, Schwann cells wrap around the elongating axon as it makes its way distally towards the effector organ. Unfortunately, central nervous system axon regeneration is extremely limited for the following reasons. Oligodendrocytes secrete growth-inhibiting molecules instead of growth factors. A large number of axons crowd the central nervous system, complicating regrowth. And regrowth is obstructed by scars formed by astrocytes and connective tissue. Multiple sclerosis is an autoimmune disease in which oligodendrocytes are attacked by immune cells. Myelin sheaths of the central nervous system are destroyed and reduced to non-functioning, hardened lesions called scleroses. Neurons cannot transmit information because of the lack of insulation. There is no cure, but the disease can be managed. Common symptoms of multiple sclerosis include visual disturbances, problems controlling muscles, speech disturbances, and urinary incontinence. Multiple sclerosis destroys oligodendrocytes in the central nervous system, leading to the demyelinating of the neurons. It is an autoimmune disease, and the pathogenesis is not fully understood. It may be a mix of genetics and the environment. Some remyelination can occur, but after repeated attacks, a scar-like plaque begins to form around the damaged neurons. The life expectancy is the same as in unaffected individuals. There is no cure, but this disease can be managed. This illustration shows a healthy neuron on the left and a damaged neuron from multiple sclerosis on the right. Affected areas include vision loss, loss of communication along motor pathways in the brain and sensory pathways as well. Intelligence is not affected. Spinal cord, disturbed speech patterns, indigestion, urinary incontinence, loss of sensation, and muscle movement. Now let's look at nervous tissue physiology. This will be similar to what we learned about with action potentials in the muscle tissue lectures. Neurons and muscles both conduct electrical energy by moving ions across the cell membrane. Here are some terms associated with electrical energy: voltage, current, and resistance. Voltage is the measure of the amount of difference in electrical charge between two areas. It reflects the distribution of ions. Recall, the ions are sodium and potassium. Current is the movement of ions across the cell membrane. This is the electrical energy harnessed to do work. So when we talk about the movement of sodium ions to the inside of the cell, or movement of potassium ions to the outside of the cell, that is the current. That is the electrical energy harnessed to do work. Resistance is the opposition to the movement of ions across the membrane. The cell membrane provides resistance to the movement of ions. Resistance decreases when protein channels open. The sodium-potassium pump helps to establish and maintain this resistance. Several types of protein channels facilitate the diffusion of ions: leakage channels, sensory-gated channels, chemical-gated channels, and voltage-gated channels. Leakage channels remain open, allowing ions to diffuse freely. Sensory-gated channels open in response to mechanical stimulation. Chemical-gated channels open in response to neurotransmitters. And voltage-gated channels open in response to voltage change. Leakage channels remain open. The particular electrical properties of certain cells are modified by the presence of this type of channel. There are more potassium leakage channels than sodium leakage channels. Therefore, potassium generally exits the cell at a greater rate than sodium enters. This continued exit of potassium from the cell helps to establish the resting membrane potential of negative 90 millivolts in a muscle cell. Mechanical gated channels will be opened when the extracellular surface of the channel protein is stimulated by mechanical pressure or vibration. This opens the pore to allow selected ions through. This is one of the major ways that tactile sensory neurons detect stimulation of touch. Chemical or ligand-gated channels will open when a neurotransmitter binds to a specific location on the extracellular surface of the channel protein. In this case, we see acetylcholine binding to a chemically gated channel. When this occurs, the pore opens to allow select ions through. We will see the inflow of sodium ions and an outflow of potassium ions. This helps to bring us to our localized depolarization, or our threshold, that will then initiate the action potential. Voltage-gated channels are opened when the voltage changes around them. Amino acids in the structure of the protein are sensitive to voltage and cause the pore to open to the selected ion. In this case, this may be a potassium channel, and for instance, at a voltage of negative 70 millivolts, this voltage-gated channel remains closed. But if the voltage reaches negative 55 millivolts, the channel opens and potassium can exit the cell. The interior of the neurolemma, or plasma membrane of the cell body, or the neuron, maintains a resting membrane potential, or RMP, of negative 70 millivolts. So notice that the resting membrane potential of a neuron is not as negative as the resting membrane potential of a skeletal muscle. The interior of the neurolemma has a negative charge relative to the exterior. This polarization of charges represents the voltage. Movement of ions across the neurolemma conducts an electrical current. Potassium ion diffusion is the most important factor in establishing the resting membrane potential. Potassium ions diffuses out of leakage channels at a greater rate than sodium ions diffuses in. Large protein anions, or negatively charged ions, that are attracted to potassium also contribute to the resting membrane potential. The leakage of potassium ions out of the cell, coupled with the proteins left behind, brings the resting membrane potential to negative 70 millivolts. The sodium-potassium pump maintains resting membrane potential at negative 70 millivolts by actively exporting 3 sodium ions and importing 2 potassium ions. This results in a higher concentration of sodium outside of the cell and a higher concentration of potassium inside of the cell. This concentration gradient allows for the generation of an action potential, which reverses the resting membrane potential from negative 70 millivolts to a positive 30 millivolts. During this process, sodium ions will rush into the cell during depolarization. The sarcolemma, neurolemma, or plasma membrane of the neuron, will become positive 30 millivolts. Potassium will flow out of the cell during repolarization, and the neurolemma will return to its negative value. And this illustration shows the neuron cell membrane, or neurolemma, at resting membrane potential. We have more potassium leakage channels than sodium leakage channels. Therefore, more potassium ions will exit the cell than sodium ions will enter. This increased exit of potassium ions will take positive charges out of the cell membrane, bringing it towards a negative value. Negatively charged proteins that are attracted to the potassiums will also be left behind because they are too large to leave or exit the cell. The loss of potassium ions and the clustering of negatively charged proteins will establish our resting membrane potential to negative 70 millivolts. We also see several sodium-potassium pumps that are going to maintain the resting membrane potential by continuing to pump two potassiums back into the cell and removing 3 sodiums from the cell. The plasma membrane of a neuron is called the neurolemma. There are 4 functional segments of the neurolemma. The receptive segment includes the dendrites and cell body. The initial segment is commonly called the axon hillock and connects the cell body to the axon. The conductive segment runs the length of the axon, and the transmissive segment includes the synaptic knob and contains neurotransmitters. So we're now going to look at each segment of the neurolemma or plasma membrane of the neuron: receptive segment, initial segment, conductive segment, and transmissive segment. This will take us through the rest of our chapter, because as we get to the transmissive segment, then we'll talk about some neurotransmitters. This illustration shows the 4 segments of the neurolemma. The receptive segment is responsible for the binding of neurotransmitters released from presynaptic neurons. It also produces graded potentials. The initial segment, or axon hillock, summates the graded potentials. This initiates the action potential if the summation causes depolarization of this area. So this is where the threshold must be reached in the initial segment. Reached. The conductive segment is responsible for the propagation of the action potential. And the transmissive segment contains the neurotransmitters, and the action potential causes the release of the neurotransmitter from the axon terminals. Graded potentials are small, short-lived changes in resting membrane potential that occur on the receptive segment of the neurolemma. They are established in response to neurotransmitters. They conduct short-lived currents, and a large stimulus opens more channels, allowing more ions to flow. Here we see the resting membrane potential at negative 70 millivolts. The graded potentials in green are excitatory. The graded potentials in red are inhibitory. Graded potentials will summate or be added together, the excitatory and the inhibitory, and if they end up depolarizing the membrane to the threshold, in this case of negative 55 millivolts, then voltage-gated channels will open in the initial segment and the action potential will be initiated. Thus, graded potentials can excite or inhibit the neurolemma. Excitatory postsynaptic potentials are abbreviated EPSP, and they depolarize the membrane by opening chemically gated sodium channels. Inhibitory postsynaptic potentials, abbreviated IPSP, hyperpolarize the membrane by opening chemically gated potassium or chloride channels. Neurons can receive both excitatory postsynaptic potentials and inhibitory postsynaptic potentials simultaneously. Here we see a neuron connected to several presynaptic neurons. In step 1 of the generation of an excitatory postsynaptic potential, neurotransmitter released from the presynaptic neuron binds to the postsynaptic neuron receptors, which are chemically gated channels causing them to open. So this is the neurotransmitters from the presynaptic neuron binding to chemically gated channels on the postsynaptic neuron. In step 2, these channels are opened and sodium ions flow into the neuron faster than potassium ions flow out. In step 3, the inside of the neuron becomes more positive, and this is the excitatory postsynaptic potential. In step 4, the excitatory postsynaptic potential moves towards the initial segment. So we've gone towards the threshold. But just one is not enough to bring us to threshold. Now let's look at the generation of an inhibitory postsynaptic potential, which takes us away from threshold. It hyperpolarizes our membrane. So in step 1, the neurotransmitter released from the presynaptic neuron will bind to the postsynaptic neuron receptors, and these are either chemically gated potassium channels or chemically gated chloride channels, and this causes them to open. In step 2, either potassium flows out of the cell or chloride flows into the cell, depending on the type of channel stimulated. In step 3, the inside of the neuron will become more negative, and this is the inhibitory postsynaptic potential. In step 5, the inhibitory postsynaptic potential will move towards the initial segment, moving it away from threshold. Summation adds the excitatory and inhibitory postsynaptic potentials together at the initial segment of the neurolemma. There are two types of summation: spatial summation and temporal summation. Spatial summation occurs when multiple presynaptic neurons send excitatory postsynaptic potentials and inhibitory postsynaptic potentials. The reason it's called spatial is because multiple presynaptic neurons can be connected to many different areas of the postsynaptic neuron. Temporal summation occurs when presynaptic neuron sends multiple excitatory or inhibitory postsynaptic potentials. So this is temporal because it's one presynaptic neuron that is sending different potentials over time. Temporal is time. So we have space. So we have many neurons connected to different areas of the postsynaptic neuron. And we have time, which is one presynaptic neuron sending multiple signals over time. The sum of presynaptic signals may or may not reach threshold. Typically, threshold of the initial segment is negative 55 millivolts. If threshold is reached, voltage-gated sodium channels open and an action potential is generated. Wow, this is an intense illustration, isn't it? But it's really showing some rather simple concepts of spatial summation and temporal summation. So here we see the green illustrates the excitatory postsynaptic potentials, and the red illustrates inhibitory postsynaptic potentials. In spatial summation, different presynaptic neurons initiate postsynaptic potentials within a narrow period of time. Here we have P1, P2, and P1 plus P2. So notice that P1 and P2 are connected to different areas of the postsynaptic neuron. P6 and P7 added together will have a higher inhibitory postsynaptic potential. And notice that they are on even opposite sides of the cell body. A simultaneous P1 that is stimulatory and P6 that is inhibitory can cancel each other out. So that is spatial summation. In temporal summation, the same presynaptic neuron initiates postsynaptic potentials rapidly within a narrow period of time. So P1 can send signals, okay, and they will add together to bring us towards our threshold. Or, in the case of P6, which is inhibitory, it is sending inhibitory signals one after the other, taking us away from threshold. Here's another illustration showing the cancellation. As we showed earlier, we have, like in spatial summation particularly, we had a excitatory and an inhibitory cancel each other out. And then here's in spatial summation, here's P1 and P2, and they're both sending excitatory stimuli, take us to the threshold. And then in temporal summation, we have P1 shooting multiple signals, and this is an excitatory one, so we are coming towards threshold. So that's pretty easy to visualize. Now let's talk about the conductive segment. Depolarization occurs when voltage-gated sodium channels are opened by the threshold. The influx of sodium ions brings the conductive segment of the neurolemma to a positive 30 millivolts. Repolarization occurs after the depolarization wave passes. Sodium ion gates close and voltage-gated potassium channels open. Eventually, the neurolemma hyperpolarizes and becomes more negative than the resting membrane potential. The resting membrane potential will be reestablished by potassium ion leakage, and then it will be maintained by the sodium-potassium pump. I'm going to show you this video of living neurons firing because there is no sound. The reason that they lit up is because of the current that was being created when the ions flowed across the membrane during depolarization. So when that sodium flowed in, we saw the neuron light up. This illustration shows a graph depicting the action potential. So here we are at negative 70 millivolts in a neuron resting membrane potential. So remember, it's a little bit less negative than a resting muscle. As a result of the summation of our graded potentials, if we reach negative 55 millivolts, which is our threshold here, depolarization will occur. During depolarization, the threshold was reached, voltage-gated sodium channels open, and sodium ions enter the cell rapidly, reversing the polarity from a negative 55 millivolts to a positive 30 millivolts. Repolarization occurs due to the closure of voltage-gated sodium channels and the reopening of voltage-gated potassium channels. Potassium ions move out of the cell and polarity is reversed from positive to negative. In hyperpolarization, or step 5, the voltage-gated potassium channels stay open longer than the time needed to reach the resting membrane potential. During this time, the membrane potential is less than the resting membrane potential. So we're up to a negative 80 millivolts here. And then in step 6, voltage-gated potassium channels will close and the plasma membrane will return to resting conditions by the sodium-potassium pump and the natural leakage of potassium out of the cell. This graph shows the negative 70 millivolts and the negative 55 threshold. So when you're answering your study guide questions, negative 70 millivolts is the resting state of the neuron. Negative 55 millivolts is the threshold of the neuron. So it's different than the muscle. So here we see the action potential where we have our depolarization wave followed by our repolarization wave and our hyperpolarization, and then we get back to our resting state. During depolarization, the yellow line shows that sodium permeability has increased. Sodium is flooding into the cell during this time. Once positive 30 millivolts has been reached, the sodium channels close and potassium channels open. And now we see an increase in potassium permeability. Potassium is leaving the cell, exiting, and that loss of positive ions is then going to bring us back to our negative membrane potential. We're going to continue to lose potassium though and hyperpolarize. This signals the potassium channels to close. And through the sodium-potassium pump and leakage channels, we will reestablish our resting membrane potential of negative 70 millivolts. The refractory period is the time after the start of an action potential when it is impossible or difficult to initiate another action potential. We have the absolute refractory period and the relative refractory period. The absolute refractory period occurs first. Voltage-gated sodium ion channels are open and then become inactivated. No stimulus can initiate another action potential during this time. This ensures propagation goes towards the axon terminal. The relative refractory period occurs just after the absolute refractory period. Another action potential is possible because voltage-gated sodium ion channels have reset. Some voltage-gated potassium channels are still open during this time, and the cell is slightly hyperpolarized and further from the threshold. Thus, the minimum stimulus strength is now greater for the threshold to be reached. That is why it is more difficult. So during the absolute refractory period, no stimulus can initiate another action potential. And I like to think of it as the action potential is in process. We're depolarizing and repolarizing, and thus during that actual process, we cannot have another stimulus initiate another action potential. But before we get to our resting state, we could have another stimulus come in, if it's strong enough, to go ahead and initiate us into another action potential if it can bring us up to threshold. That is our relative refractory period. Two types of action potential conduction can occur. We have continuous conduction and saltatory conduction. Continuous conduction occurs on unmyelinated axons. Saltatory conduction occurs on myelinated axons. This is going to explain why myelin is so important. During continuous conduction, the action potential will open voltage-gated channels continuously along the axon. This results in a slower conduction speed. In saltatory conduction, the action potential occurs at nodes of Ranvier, where voltage-gated channels are concentrated. The action potential will travel through the myelinated segments of the axon and become weaker, but they will remain strong enough to open voltage-gated channels at the next node. The process repeats itself and the impulse jumps from node to node. This is much faster than continuous conduction. Illustration A shows continuous conduction, and you can see that the channels have to continuously be opened as the depolarization wave travels down the unmyelinated axon. Whereas during saltatory conduction here in illustration B, The voltage-gated channels are concentrated at the nodes of Ranvier. The action potential can proceed undisturbed through the myelinated region of the axon. It does lose its strength, but it still has enough of a depolarization wave to open sodium-gated channels, voltage-gated channels in the next node of Ranvier and reinitiate the next action potential. And so it basically hops. So whereas we had to move very slowly across an unmyelinated axon, in a myelinated axon, we can hop that same distance in a fraction of the time. The synaptic knob contains the transmissive segment of the neural lemma. At the synaptic knob, the action potential will open voltage-gated calcium channels. The calcium ions diffuse into the synaptic knob and binds with synaptic vesicles. This triggers exocytosis of the neurotransmitter into the synaptic cleft. The neurotransmitter binds to chemically gated receptors on the target cell, initiating a physiological response. This illustration shows the events of the neuromuscular junction as learned about in the last chapter. In step 1, the nerve signal or action potential reaches the synaptic knob, where it opens voltage-gated calcium channels. Calcium will then enter the synaptic knob and bind with proteins associated with the synaptic vesicles. The synaptic vesicles are filled with neurotransmitters. They will fuse with the synaptic knob plasma membrane, and the neurotransmitter will be released through exocytosis. In step 4, the neurotransmitter will diffuse across the synaptic cleft and attach to the receptors on the muscle. So in this case, the neurotransmitter is acetylcholine. This will then open the receptors on the neuromuscular junction. We learned that it was the junctional folds at the motor end plate, and we will reach our threshold for the muscle, which is negative 65 millivolts. All right, so the synapse is going to be where we talk about our neurotransmitters as promised. So in order to effectively communicate with other parts of the body, neurons synapse with other neurons, muscles, and glands. So neurons can stimulate or inhibit other neurons. As far as muscles go, neurons always excite skeletal muscles. Neurons may excite or inhibit cardiac and smooth muscle. For glands, neurons will stimulate the release of hormones or exocrine substances such as sweat. This video on how neurons communicate has sound, and thus I will not show it, but the link will be posted. This illustration shows the 3 types of synapses. Neuron 1 synapsing to neuron 2, so that's neuronal connection. Here is a neuron synapsing with a muscle, so that's a neuromuscular junction. And then we have a neuron synapsing with a gland, so that's a neuroglandular junction. The chemical synapse is the most common type of synapse. Here are the features of the chemical synapse: the presynaptic neuron, the synaptic knob, the neurotransmitter, the synaptic cleft, chemically gated receptors, and the postsynaptic membrane. So as we know, in step 1, the nerve impulse reaches the synaptic knob where it opens the voltage-gated calcium channels. Calcium flows in and binds to synaptic vesicles containing the neurotransmitter. Synaptic vesicles then fuse with the plasma membrane of the synaptic knob and releases the neurotransmitters through exocytosis. The neurotransmitters then diffuse across the synaptic cleft and bind with the chemically gated receptors on the postsynaptic membrane. There it opens those receptors and allows for the inflow of sodium ions into the cell if it is an excitatory neurotransmitter. Neurotransmitters are the chemicals that transmit information within the nervous system. More than 100 neurotransmitters have been identified and organized into different groups. Neurotransmitters stimulate a physiological response in the target cell. Most neurons make two or more and may release any one or all of them. Most neurotransmitters are released at different stimulation frequencies, which is what helps make this a precise system. There are four main chemical classes of neurotransmitters: acetylcholine, biogenic amines, amino acids, and neuropeptides. The acetylcholine structure will differ substantially from other neurotransmitters. For the biogenic amine, an amino acid is slightly modified to synthesize the neurotransmitter. These include catecholamines and indoleamines. Catecholamines are made from tyrosine and include dopamine, epinephrine, and norepinephrine. Indoleamines are made from tryptophan or histidine, and they include serotonin. Amino acids include common neurotransmitters such as glutamate, glycine, and GABA. Neuropeptides are chains of amino acids up to 2 to 40 amino acid long and include endorphins. So here we see some structures of common neurotransmitters. We have the adrenaline and noradrenaline and dopamine. These are biogenic amines. Epinephrine and norepinephrine are in that class of adrenaline. And then we have dopamine. These are all the catecholamines made from tyrosine. Adrenaline is the epinephrine. Noradrenaline is norepinephrine. And dopamine. These are your catecholamines made from tyrosine. And then we learn that serotonin is a biogenic amine, or called an indoleamine, and it is made from tryptophan. Turkey has a high concentration of tryptophan, which may be one of the reasons why it makes you sleepy when you eat it. Here is acetylcholine, which is in a class of its own. And we had our amino acids, which were glutamate, glycine, and GABA. So here's GABA and glutamate. So these were amino acids themselves. And then we had neuropeptides, which were chains of amino acids and included the endorphins. So there's your endorphins. And this illustration will describe the effects of each of these different neurotransmitters, if you'd like to go back and read them. So that was the structure. Neurotransmitters are also classified by function. Classification is the effect on the membrane potential, whether it's excitatory or inhibitory. So excitatory transmitters will cause excitatory postsynaptic potentials to fire, and inhibitory transmitters will cause inhibitory postsynaptic potentials to fire. Some neurotransmitters can excite some targets and inhibit others, depending on the specific receptor. We'll learn more about that as we get to the autonomic nervous system with the parasympathetic and sympathetic nervous systems. Removal of neurotransmitters from the synaptic cleft can occur in several ways. Enzymes might degrade the neurotransmitter. We saw that with the acetylcholinesterase in the neuromuscular junction. Reabsorption of the neurotransmitter into the presynaptic neuron can occur, and this is called reuptake. And diffusion away from the synapse and being engulfed by a neuroglial cell, such as microglia or an astrocyte. Those are the ways that neurotransmitters can be removed from the synaptic cleft. Some drugs have their effect by influencing transmitter removal. For example, Selective serotonin reuptake inhibitors can treat depression. For example, galantamine hydrobromide is an acetylcholinesterase inhibitor used to treat Alzheimer's disease. So those are two different medicines. Selective serotonin reuptake inhibitors treating depression, and galantamine hydrobromide acetylcholinesterase inhibitor treats Alzheimer's. Acetylcholinesterase is very important in cognitive function. We'll discuss that in a later chapter. Neuronal pools, also known as neuronal circuits, are groups of neurons arranged in specific patterns. There are 4 types of circuits: converging, diverging, reverberating, and parallel after discharge. The main thing to know about this is that with our circuits, the brain can send several types of messages using the neurotransmitters and voltage and pathways as they travel to and from different parts of the body. Converging circuits occur when several neurons synapse with a single neuron. Thus, many inputs will result in one output. So that's going to be very important with like sensation. Diverging circuits occur when one neuron synapses with several neurons, and this is going to result in many outputs. We saw that with a motor unit where one motor neuron synapses with like several muscle fibers. So they can do that with neurons too. Reverberating circuits occur when neurons connect in a feedback loop, and an input causes a cycle which will run until it's turned off. And that helps you to continue to beat your heart and breathe without thinking about it or taking unnecessary energy. And then parallel after discharge circuits occur when neurons send information along different pathways. Input results in outputs that continue after the input has stopped. So this is a video explaining those neuronal pools. Start it at minute 2:54 seconds, and I will include the link. This is a converging circuit where several neurons will converge with the output. So several inputs, one output. And we have a diverging circuit where we have one input and several outputs. We have the reverberating circuit, which keeps it running. And then parallel after discharge. So here is our input, and then we can see that the output will continue well after the input because it just kind of keeps hopping. So watch that video to further explain how each of these work. And Frank and Ernest says, wow, this says the human body has 45 miles of nerves. No wonder someone is always on mine. Thank you all. Have a good day. Be blessed.